Welcome everybody. We are live in Berkeley, California. My name is John Cumbers and I'm the founder of SynBioBeta. And today we have a deep dive technical webinar all about natural product synthesis and also using the Labcyte Echo to do liquid handling. We're here at the home of Tierra Biosciences and I'm joined by Zach Sun, who's the founder of Tierra. Welcome.
Thank you.
And Jared Bailey, who's an application field scientist at Labcyte. We're very excited to tell you about what's coming up today, and I'm just going to give you a preview of it while we're waiting for a few more folks to be joining us, and then we will dive into the technical content, which is going to be a presentation by Jared on the Echo and a presentation by Zach on cell-free synthetic biology and natural products. So these web deep dive series are focused on technical aspects of synthetic biology. They're interactive, they're live, so we do welcome your questions. Type them into the Q&A box and we will probably answer them as we go, or we'll answer them at the end of the presentation. As I said, today's topic is about cell-free and acoustic liquid handling and how it's disrupting natural product discovery. We'll talk a lot about cell-free, we'll talk a lot about the natural products that are involved, and how you can do all that while reducing consumables costs using the Labcyte Echo. So we'll be talking a little bit about some of those consumables and why the Echo is such a beautiful piece of equipment for doing that. I've often joked that with Rich Ellison, the founder of Labcyte, that the Echo should be every startup's first employee because it's so useful, and it can work 24/7 as well.
Very easily can, yes.
Exactly. And we're also going to be talking about some of the key to Tierra's high throughput approaches is using the Echo and liquid handling and lab automation to do that. We'll then conclude, as I said, with a live Q&A from the audience, and we encourage questions as we go. So I think we are going to dive right in. And again, for those of you who just joined, this is SynBioBeta Deep Dive. My name is John Cumbers. I'm the founder of SynBioBeta. And SynBioBeta is an activity hub for the synthetic biology industry. We hope that you'll join us in October for our big annual event in San Francisco, where Labcyte is one of our big sponsors of the event. And you can actually roll up your sleeves and look at an Echo in real time. And Jared has spoken at SynBioBeta for a number of years about that. So today's topic is how cell-free and acoustic liquid handling will disrupt natural product discovery. And we do it while reducing consumables. And if you missed us a second ago, we are live at Tierra Biosciences in Berkeley, California. And Tierra is bringing a cell-free approach to natural product discovery. And a key to this is using the Labcyte Liquid Handler. We will be doing live Q&A. You can type them into the box. And we'll be doing questions throughout and then also a summary at the end. So it's a pleasure to introduce Zach Sun. I've known Zach for a number of years. He's been incubated here at the Potter Street campus. And he's also part of the Cyclotron Road Accelerator, or was part of the Cyclotron Road Accelerator, which is a wonderful program that gave you your head start, part of Lawrence Berkeley National Lab. Tierra Bio recently changed its name from Synvitrobio, and Zach, perhaps you can tell us a little bit about the thinking behind the name change, and has been developing fundamental synthetic biology technology since 2006. Zach has published work on MAGE, genomic engineering in Nature, and on cell-free prototyping environments in eLife, and the latter publication forms the basis of Tierra Biosciences. And he holds a PhD from Caltech and an AB in chemical and physical biology from Harvard. So welcome, Zach.
Thank you. Thanks.
And Jared Bailey is an application scientist who's been at Labcyte for the last 2 years. During that time, he's been applying the benefits of acoustic liquid handling to various workflows within synthetic biology. And he received his master's degree from the University of Kentucky. And he previously worked at both Elixis and Intrexon on the biosynthetic production of terpenes and isobutanol. So it's great to have you as a synthetic biology expert inside Labcyte.
Inside the liquid handling company, yes.
Exactly.
It's great. They like to have the expertise in there. So.
Exactly, welcome. And Jared's showing us all up with a nice branded shirt. Okay, so Jared, just for people who are not familiar with the Echo, what is it? What does it do?
It's great. So as you mentioned before, it's an acoustic liquid handler. So it allows you to move from the microliter scale that everybody's used to in lab down to nanoliter scale. So you have 2.5 or 25 nanoliter granularity, depending on which model you have.
Okay.
So you're able to go much, much smaller in lab in much shorter amount of time, is in short.
Excellent. Well, you've prepared a few slides, so why don't you dive in and tell us a bit more about it?
Thank you very much, John. All right. So yeah, let's talk about the 25 nanoliter version, which is the most applicable to what we've been doing here. It's what Tierra has, and it's what I've been doing my cell-free work in-house with. So it's actually our large drop dispenser, as mentioned before. It dispenses 25 nanoliters, and in general terms, it quickly dispenses these drops for a flow rate of around 5 microliters per second. So that's the max rate. Depending on your fluid type, that can change, but that's that enables you to do reactions in a fast manner that you would never even be able to think about doing at the nanoliter scale by hand. Oops, sorry guys. There we go. So let's actually talk about it. How is this game changing? It's very similar when you think about an Echo. It's just going from like a 19th century horse-drawn carriage to a Tesla. And the fact that a lot of these reactions that I was doing or that I've heard of people doing before were done by hand. We're actually manually arraying the liquids. That is extremely slow, inefficient, time-consuming, and just doesn't work out very well. It breaks your lab personnel very quickly. So it's nice to be able to move to a more high-throughput setting, and the Echo is a very good way of doing that. The game-changing in lab can be visualized as this, sort of the difference between the horse carriage and the Tesla. We're going to go over these differences today as it applies to cell-free experiment that we did in lab. But one can envision the large increases in speed, automation, and precision being true for both technology upgrades as shown here. Before going over the experimental work, let's look under the Echo's hood and see how it functions and just show you sort of how the actual technology works. So we'll put up a little short video here that our great marketing team has put together. So when you actually look at an Echo, we have a bunch of different Echo qualified source plates. These come in different flavors and sizes based upon your liquid handling needs. The Echo can handle most any typical SBS format plate that's 14 millimeters in height or less. It takes that plate and inverts it. And then what you're actually seeing here is there's a transducer that actually is just like what's in your stereo speaker. That has a little bit of coupling fluid on top of it to allow acoustic energy to go through it. And we're going to be measuring 3 things. We're going to measure the bottom of the plate, the bottom of the liquid, and then the top of the meniscus of the liquid. There's a reflection or an echo, which is where the name comes from. So that's what we're measuring, and it's done in a very quick manner when we actually scan through the plate. So we can tell different fluid types. You can actually put in, we can do dynamic fluid analysis. You're able to tell how much DMSO is in a plate. There's a lot of different measurements you can actually get from this. But in short, the short way of looking at it is we're actually able to measure how high that liquid is off the bottom of the plate. So we can focus acoustic energy effectively. So you'll see right now, this is actually what happens when you put in the plate. It's scanning the entire plate and just quickly it's pinging just like a sonar ping would, and telling us how high that liquid is and all those measurements really fast. You get a lot of information from this scan. So then after it's done with this scan, it can go back to whatever well you want to transfer from and effectively focus that energy on the meniscus and transfer with the 525 case a 25 nanoliter droplet to the inverted destination plate. And that's held in place by surface tension. So that drop is then quickly transferred, and then around 400 to 500 drops per second, we can actually transfer other fluid types. And both stage and the transducer are independently moved, so you can do any well to any well. If you can imagine the plate moving and then also the transducer below the source plate moving, you can switch up any possible combination of wells. So hit picking is very, very fast with this instrument. That's one of our main advantages, actually, I would say. We're able to do that very quickly and effectively. So that's the short term of what the technology is doing under the hood.
Tell us again, Jared, what's the smallest volume of the droplet?
So we have 2 different models. We have a 2.5 nanoliter model and we have a 25 nanoliter model. And they have a little bit of a different market. For the TXTL work we've been doing, the 25 nanoliter volume is a little bit more applicable. Okay. Just because you can imagine a 10 times increase in speed. You don't need that smaller granularity necessarily. You need more of the speed of the 25 nanoliter drop.
And the TXTL is the cell-free
Yes,
applications
yes.
that we're going to hear about in a second.
Yeah, sorry. Yes, it's the typical cell-free workflow.
And let's just define that TXTL. I can't remember. It's reticulocyte. Do you know what it is?
Transcription translation.
Oh, okay.
So it's just you're putting in DNA into a system, and then you're able to actually get transcription of that DNA into RNA, and then they're able to take that RNA and actually transfer it into protein within the same system. So all of the necessary cell components are in there, except for the cell's not there anymore. They've either lysed it or they've added those components in individually. So you're able to effectively remove the cell, which has a bunch of advantages that I'm sure Zach's going to go over that in much better detail than I could. So, but actually, let's talk a little bit about one of the experiments that we've done at Labcyte. So, what we've been able to do here is take a system where we have this MyTXTL mix. We were able to work together with Tierra Biosciences for this, who makes a commercial product called MyTXTL. They provided that for us, and then what we're showing here is. I try to keep the colors the same. So the MyTXTL is arrayed in an echo source plate. There's water in there for backfilling so that we can keep the volumes the same. And then we're putting in two different plasmid A and plasmid B. And that's your DNA input for that TXTL. So that's actually the input we're putting in. And what we're going to have out of it at the very end is GFP. So we'll have a nice readout at the very end. So plasmid A induces plasmid B, which gives us GFP. So all that's being said is we're going to increase the concentration of these two different plasmids and have a multivariable experiment. That is very challenging to even think about doing by hand. These are time-dependent reactions, so it would actually be impossible. You would not be able to do this quickly enough to actually have them relate to one another, because it would be, by the time you could do P24, you would have actually, A1 would have actually moved way past where it needed to be. So you wouldn't be able to compare them to one another. So what I'm just showing in this multivariable assay plate is that difference in concentration. And what we're expecting to see is, as you would expect, hopefully, as plasmid B and plasmid A increase in concentration, you would have a requisite increase in GFP. And we'll see that in the next slide, but it's not exactly what you would imagine. So here, we were able to see the concentration differences. When you actually add them up, you're increasing plasmid A as you go from left to right, and plasmid B as you're going from bottom to top. But it's not just a simple, the red is the increase, the highest level of GFP, and purple is the lowest level. And you're not seeing red in the very top right-hand corner. There's actually a ridge that's sort of formed in the middle, if you would imagine this as a three-dimensional plot. That is due to a bunch of different reasons. It could be due to competitiveness within the TXTL resources themselves, within the reagents. There can be a concentration differential. There's a lot of different stuff that can cause that. But you would have never even seen this by hand. The ability of the echo to transfer the different fluid types at the same time quickly, and the ability of a person to actually input this kind of DNA, this kind of system in, it would be very challenging to even think about putting this in by hand. And the echo, it's a very easy input. As you saw on the previous slide, the amount of inputs you need are very low, and our software package makes it extremely easy to be able to put this kind of an experiment together. So a person can do, I did this in a single day. This would take weeks to do by hand.
So I just want to go back a second. So it's the concentration of the plasmid that you're adjusting?
Yes, the two different plasmids across the plate.
Okay, and both together are needed to make GFP?
Yes, so plasmid A turns on plasmid B,
okay,
which then makes GFP.
Okay.
So it's sort of a simple biological circuit is one of the best ways of saying it, but it's not exactly, you can't turn it on and off, but plasmid A turns on plasmid B, and then that makes GFP at the end.
Got it, okay. And I know a lot of people in the synthetic biology industry are using the echo for DNA assembly.
Yes.
And we did a whole webinar with Amyris a few months back, looking at DNA assembly. So this is great to see now the echo being applied to actually a genetic circuit, a simple circuit, and I can see all sorts of applications in terms of being able to optimize any kind of circuit you want.
Yes, and it's used, there's Vincent Nero actually is one of he's in academia. He actually uses this heavily in his lab for a lot of biological circuit prototyping. So he's done a lot of work with this, very successful.
Where's he based?
I actually forget the institution.
Minnesota.
Minnesota, yeah.
Okay. Yeah. Great.
So yes, that's the basics. And as you mentioned, it works very well for a lot of other applications in lab. It's not going to be just used in your lab for TXTL. You can use it for DNA assembly or something like NGS, next generation sequencing of the actual plasmids after you make them. That's a crucial part of any kind of lab workflow. The Echo enables both of those assays to happen, or both of those workflows to happen. And it's done through our ability to miniaturize assays is one of the biggest ways we have your return on investment from buying an Echo. You can miniaturize anywhere. I mean, typically the limit is not the Echo, it's the detection method you have at the end of your assay. That's your limit. It's not the Echo itself is what we found. So depending on your detection method, we've gone all the way down to 100x reduction in volume in a lot of assays, or even lower than that for some, but really that's a comfortable level we're at. And on top of that, you're actually going to increase your data quality from all of this stuff. Person-to-person variability is a big problem. That's a major problem in any lab with pipetting. You remove that as a variable. So you're actually able to just, a single person feeding into a robot, you now have the same results coming out of each individual's hands. This is going to also allow you to accelerate your productivity. You're allowed to, that design, build, test, learn circuit, you're able to go a lot faster around the testing part of it and the design part. You're able to feed back into your results a lot faster. Also, we're able to automate. We have automation platforms as well. If you want to go up, if you start thinking about how fast an Echo is, you actually need to feed plates in faster. So as your lab expands, we have automation that's capable of handling more plates and then feeding that into your readers of whatever they may be. And then lastly, we have compliance software. We have a bunch of different software packages that make the Echo usable for different types of labs. Like if you wanted to do dose response, if you're doing if you're testing out small molecules against cancer cells, we have that. We have this, what I use is Echo plate reformat, which is the ability to quickly input a source plate and destination plate and volumes in between. And we have a software package for most general use needs that you can buy and we can put together. This one's just mentioning we have a 21 CFR Part 11 software package if you have to be in compliance for that particular.
That would be is that something related to GMP or?
Yeah. So if you're in a drug manufacturing lab, a lot of times that locks it down so that only people, you can see whoever's logged on or whoever makes a modification to the software. So you can see any modification at all that's been done to the instrument or software. It pops up. But yeah, we have the ability to be used in a bunch of different environments.
Excellent. Well, if you've got any questions for Jared, then type them into the box. I think we're going to move over to Zach now.
Perfect.
And we'll see how you're using the Echo. And I see you were bringing up a Medium post that you highlighted. What was the title of your Medium post?
That's right. Yeah. Let me put it into the webinar chat here. But for those of you that are curious, I think there's a lot of different applications of cell-free biology. I wrote up recently a post kind of outlining what I was really excited about. But in there are some applications that, some that we've seen already, but also some that are kind of new, kind of on the horizon for this technology. So I think most of which Echo can help. So
the title is Why I Believe in Cell-Free Biology. And so if we can bring up Zach's slides, then we will move over to your presentation. And I have to say, I think that I've said this to you before, Zach, that when I first heard about the term cell-free biology, I thought it was cheating.
Yeah.
I thought, hey, the beauty of biology, one of the benefits of biology is the fact that it's self-replicating. And so I thought you're trying to do cell-free synthetic biology. That's taken the challenge out of trying to engineer biology.
Right.
And then when I kind of just got knee deep in trying to engineer biology, and you realize how difficult it is, then I was like, okay, please, thank you. That's so nice. Just get rid of replication and all the stuff that the cell does and everything it turns on and off during cell division. So what got you into cell-free synthetic biology in the beginning?
Yeah, it's a good question. So I was lucky enough to have Vincent Moreau on my thesis committee.
Oh, great.
And he really introduced me to this field. What was fascinating to me was I'd spent a lot of my early PhD just engineering cells. And it's just this laborious process, right, of doing design, build, test. You wait a couple of weeks, your plasmids might clone, it might not, and then you get a result. And you're just one data point, and if it didn't work, you just kind of do it over again, right? And for me, I said, well, cells are really great. They replicate. They're great vehicles for making things. I don't think anyone can question that. But is there a way that we can go faster, kind of look more like engineering instead of just design, build, test, so that we can get to our end result, which might be a cell, right, that's making chemicals faster. And I think that's what really, got me thinking, well, this technology, it might be a fundamental technology that we can kind of base this field on, right? I mean, there's circuit prototyping applications, there's identifying novel chemical matter applications, but really, it's just a fundamentally different way of looking at engineering biology, right? And it kind of ties really nicely into kind of cellular formats of engineering. I think that's really exciting for me in this field.
Who invented the idea of doing cell-free?
So cell-free is actually a really old idea. Many people don't know this, but in 1968, the discovery of the amino acid code was done in cell-free systems, and a Nobel Prize was given for it. So it kind of predates a lot of the cellular ideas. Before we could manipulate cells in the 80s, this was the only way we could really understand how nature worked. And so it's a very, very old idea, even older, than almost discovered DNA.
Cool. Well, let's dive in. I've got a lot more questions and comments on why 2019 is the year of cell-free.
Sure.
I think I said 2018.
2018?
But I'm just pushing it out a year because it continues to grow in excitement. And we have a whole session on cell-free synthetic biology at the conference in October. And you mentioned Tierra Biosciences. They're a new sponsor for this year as well. So there's a lot of cell-free going on. So tell us about Tierra. Tell us about discovering molecules from nature.
Sure, yeah, absolutely. Great. So Tierra, we formerly were known as Synvitrobio, which stood for synthetic in vitro biology, but we wanted to generalize the company to really focus on what we think we're really good at. And one of those things that we're really excited about is discovering new molecules as potentially like a molecular search engine to find new chemistry out there that exists in biology. And so it's really driven by this idea here. Penicillin, I think most of the audience knows, a drug invented in 1928 was really the first time we could beat infectious disease. And all of the drugs henceforth that are antibiotics have really changed the way that we treat patients. This came from a fungi. This was very much a natural product. And what that means is it existed in nature, but we had yet to discover it until 28, right? But what the audience might not know is other drugs such as Lipitor, which is a best-selling cholesterol drug, right? Is also from a bacteria, and so this is also a natural product. And you can even zoom out kind of outside of the therapeutic world. Glyphosate, which is better known as Roundup Ready, is yet again another natural product that originates from bacteria that's really changed the way that we do agriculture, right?
Does that make it organic then?
Right, and now there's organic natural products too, right, that are coming out that are kind of new pesticides too. So yes, there will be eventually. But so when we look at nature, right, nature's been this fruitful source of natural products, and we take a pretty liberal view on this, right? From Roundup Ready to Lipitor to antibiotics, even chemotherapies. But I don't have to tell you about kind of the antibiotic resistance crises we've had or like the lack of finding new herbicides and pesticides. We really haven't had much luck in finding kind of new natural products. And when I think about that, at first when I learned that, I was pretty surprised. Is it just because with all the new technology, we're just not looking hard enough, not looking in the right places? But indeed, I was fascinated to learn. When we think about the genetic space that we've actually explored using all the technology we have, we've really explored less than a pixel in this whole slide. And what I mean by that is here I have kind of a wheel of different life forms, right? From plants, animals, protea, archaea, and such. And when you kind of zoom in on this wheel, what I've highlighted here is E. coli. And E. coli, is our standard workhorse of biology. I'm sure, most biologists kind of learn how to use this as a starting tool. But it really represents just a really small fraction of the biochemical diversity that we know exists out there. There's 1 trillion organisms that we know exist on this planet. We routinely grow 10,000. We routinely work with 10, such as E. coli. And so there's all of this space that has not been explored, right? And probably contains the natural products that we need to drive future society. And that's what really excites us, right? For us, we see using cell-free systems as a way where we can access this chemistry, access the stuff that we know, has useful molecules or enzymes or such, but that we just can't touch currently. And when we think about kind of cell-free versus cell, Jared did a great introduction on cell-free and how we can use it. Cell-free at its core is literally, as we see it, just a cell that can do complexity, but can do it faster by taking away kind of the cellular wall, taking away its ability to grow, right? And instead what you're left with is effectively a reagent that's able to take in different types of DNA and then do what a cell would have done anyways with it, right? Make the products that DNA would encode. And so when we think about kind of cells and cell-free and how you compare the two, for us, using cells to engineer things can take a while. You need to clone things, so you just kind of fit the requirements of a cell to manipulate. Whereas in cell-free, it's a pretty fast process. You can rapidly use it. DNA can go in as linear cassettes and be used directly. The second part of kind of cellular engineering is, you have to get the plasmid into a cell. You have to be able to grow the cell has to survive. If stuff is toxic, the cell's not going to be super happy with that. You also, are limited to kind of single strains and single points. And in cell-free, what we see, since these are reagents now, cell-free systems are, fairly easy to work with. And you can kind of emulate lots of different strains, not just one, like E. coli. And at the end of the day, cells give you one data point, and cell-free can give you hundreds of data points at a lot cheaper cost. This is not to say that cells don't have a use, right? For making chemicals, indeed, like the ability for a cell to self-replicate, I think is a huge asset, right? But can you use cell-free systems to even help you in that process, right? For us, cell-free systems are a data collection model that really show you, what is it? Can you understand your systems better? And furthermore, can you identify kind of natural products from there?
So if you think about the synthetic biology, the design build cycle,
right?
The design build test cycle,
right?
Is cell-free just in the test part?
Yeah, I think cell-free plays a really large role in the test part, but also plays a large role in the learning part, right? So then you can go back to design build. It influences design and build because your requirements in cell-free are, I'd say, less stringent than in cells, right? So instead of having to clone things during your build part, you can just use kind of linear assemblies of DNA, linear DNA, and that makes it go faster too. Yeah. Great.
We did have one question that came in from Dongwon Park. He says, what's the biggest protein that you could make with the Echo, I guess with a cell-free system? Do you have any limits, or is it whatever nature can make, you can make in a cell-free system?
That's a good question. A lot of cell-free biology hasn't been super well characterized as of yet. So I believe there's been some literature making kind of CRISPR-Cas proteins in cell-free, and that's a pretty big I think that's like 9 kb or something. So that's pretty big already. But I think it's still kind of a question in progress if we're trying to understand.
Great.
In the Echo limitations, there's not really on that end. We can transfer the DNA needed for that, whatever plasmid size. The limitation would be on the TXTL.
On the biology itself, yeah. Exactly, right.
Great.
So, so this is kind of a picture of what we typically run things in, which is 34-well plates, kind of sitting on a Labcyte Echo tray, right? And so each of these kind of represents a different type of reaction that we'd run. But just, our typical workflow involves kind of setting up these reactions and these microfermentations, running them for 8 hours, and then seeing kind of what shows up. And I think, one of the things, we're a startup. We're 10 people, but after we raised our seed round, the first purchase we knew we had to make was a Labcyte Echo, and I think that speaks a lot to how useful the instrument has been for us. What I have here on the left is kind of when we first started using the instrument, we had access at Caltech to one, and so that's a 6-hour drive, maybe 5 hours flying to LAX and a delay, right? But it was that useful for us that we took that to use it. Recently, Labcyte installed an Echo at QB3, and it was super exciting for us because now instead of that drive, it was cross the bridge during rush hour, right? But even then, that really just couldn't fulfill our needs, and so that's when we knew we needed to get a Labcyte Echo to really drive our research forward. Yeah.
Can I just go back a second to when you saw the dots? The actual fermentation is done on the slide. You said these microfermentations. So they're actually done on the slide.
That's right. Or cell-free expressions. Yeah. So these are. This is just kind of a visual to show what it would be like, but these would be in the plate.
Okay.
Yeah.
So you put it on, and then how long would that take to run, did you say?
It can take 8 hours or so.
Okay.
It can be as fast as 4 if you want to do that.
I ran mine for 16
Yeah.
overnight. You can run them, it depends on how long you're.
And you must get some evaporation because they're so
Right.
small.
So we're just showing this as kind of demonstration. This is a good point. What would actually happen would be these droplets would be within the plate, and then the plate would be sealed. So
okay.
Yeah, we actually sell. Oh yeah, we actually sell a microclimate lid that I use for that in my assay as well. That's,
Okay.
It hydrates. It makes a hydration curtain around so that you don't have.
Nice.
A lot of the reactions require oxygen to be involved, so you have to have air as well. So.
Right.
Got it.
Yeah. Good question.
Yeah.
So, part of the thing about cell-free is that different types of cell-free systems, depending on how you make them, will have different outcomes. And then furthermore, depending on how you run them, right, this kind of a user error is really pervasive in the field. When you're thinking about mixing like 10 microliter type reactions, when you have multiple components going in, unlike a PCR where it's pretty like lax on, if you have 10-fold less or 10-fold more primers, in a cell-free reaction, it's really sensitive to these parameters. And so for us, we played with traditional liquid handling, and we played with the Echo, and it was really kind of night and day what the Echo could do relative to traditional liquid handling. What I'm showing you here is kind of work that we've done where we've built a circuit, much like the circuit that Jared's shown. This one is kind of a 10R repressor that gets lifted by an inducer like ATC. And what we can show is that with different kind of preparations and then running it, that we're able to get really low CVs. And that kind of makes this technology repeatable, reliable, scalable, which is something that historically, cell-free systems haven't been great at. And so getting there kind of, we hope, advances the field forward. But really, the Echo's helped us do that. And so when we think about kind of our platform for natural product discovery, right, it starts with different proprietary libraries that then can be tested in cell-free systems. Things that are valuable, right, you can then scale, or you can take that data and move it into a cell. We're pretty agnostic as to how that goes. And then you can use cell-free to really inform learning cycles to kind of move your discovery process forward.
Great. We have a lot of questions coming in, so let's do them as we go, if that's okay with you.
Yeah, absolutely.
So a lot of questions about evaporation, which I think we've now we've answered in terms of, you said you sell a piece of equipment that
Yeah, a lid. It's actually called a microclimate lid that we use. We use it for either DMSO hydration, or inversely, you can use it for the other way. We can try to keep water in the system with adding water to it as well.
Do you use that when you run the system too? Or
yeah, I did bottom reads actually. So I did a bottom assay read, and I was able to get very clean results.
Okay.
So you don't have to read through the felt. You can actually read through the bottom.
Okay, things that we'll have to try that. We have to try that.
Neat. Yeah,
that's pretty neat. Yeah, that's great. Cool.
Great. And then we had a question from Reich. I presume that's Reich Grunberg. Hi, Reich. How large are those reactions in the end, volume-wise? I guess the ones that you're running.
Right. So we can run anywhere from 5 to 10 microliters. You can run this stuff up to 200 liters if you want. Right. 200 liters is gigantic fermenter scale. I think you guys have done even smaller than that.
I did 4 microliters, and I just wrote a tech note actually on that at G129. So it's on our website if you want to come take a look at it, if anybody out there wants to. But we got down to 4 microliters effectively.
Great.
But that's for a nice blazing molecule like GFP. Some of the ones you have are going to be more challenging, right? So that's a little bit, they have to go up for that.
Right.
So David and Antoine, I think we've answered your questions, but if you want to clarify, you can keep going. Okay, back to you, Zach.
Great. Okay. And so fundamentally for us, right, we've really spent a lot of time with help from DARPA kind of building new cell-free systems that are not just E. coli based. And that's really critical if we're going to explore kind of new chemical diversity. And each system, a feasibility reference system, the newer systems are easier, we think, to build because you kind of learn a lot from your reference system. And each system, gives you different parameters that might be useful in the process of natural product discovery. And this is just showing kind of the systems that we have built and kind of how that, how you can get benefits from each. That kind of goes into some of the work that we've been able to do kind of in this field. That I'm able to talk about. And so in this example here, we're looking for new drug-like molecules from nature. In specific, it's a cyclic peptide that can act as an RNAP inhibitor. What that means is that when this cyclic peptide is formed, it inhibits transcription. This is nice since we can see it, but also kind of that's the feature of the drug-like molecule. These are encoded by 3 genes, right? So 3 pieces of DNA that you would, if you were going in a cell, you clone down to a plasmid, but for us as linear DNA, we can just rapidly kind of screen it through. And what I'm showing you here is that really the assay here is inhibition of RNA polymerase, and so we can run a bunch of clusters that we know make cyclic peptides that inhibit, but we can also kind of run novel clusters too that inhibit that we think might inhibit, right? And so here, the Echo and Beta do really high throughput cell-free system kind of work allows us to identify kind of new cyclic peptides or RNAP inhibitors in a way that you really wouldn't be able to do in cells really efficiently. And then, based on this, we can say, okay, is this interesting? Should we make it in cells because cells grow? Should we make it in cell-free or such? But really just going to show that we can rapidly explore kind of new chemical space. Another thing that we've been doing, and this is through NSF support with UW-Madison, is to build out a system proto vitro to do kind of enzyme screening and engineering. This just shows that for difficult enzymes in a pathway, or if you'd like to engineer an enzyme to perform a new function, one way you can do it is using cell-free systems. Here we have a cytochrome P450 that takes limonene and converts it to byproducts. And we can just show that you can rapidly express different variants of this cytochrome that then have activity against limonene. But you can also express these genes and show that they're expressed by running gels and such. And really rapidly kind of explore sequence space in ways that you wouldn't be able to in cells due to toxicity or to speed.
So proto vitro is your product?
One of our products, yeah.
Got it. Okay.
That's right. Yeah.
And if you could just clarify, what's your secret sauce? What's your IP? Is it on the discovery side? Is it on the technology for the cell-free expression?
Right. I think a lot of the cell-free work kind of forms the basis of the company, so I'd probably say the cell-free area.
Okay.
Yeah.
Great. We have a couple of other questions. Are there any heating problems that you encounter with transducers?
Any heating problems with transducers?
I can speak to that.
Okay.
Are we talking about a biological transducer here, or?
Yeah.
The Echo probably would be what it would be. So that's the actual way that the sound energy is transferred through.
Got it.
We have coupling fluid that's actually kept at a constant temperature. That allows you. We have a cooling unit that sits on the side of our instrument, so that keeps the coupling fluid at a constant temperature. That keeps the speed of sound the same, and it also helps with heating issues. There's no heating issues at all that we have.
Great. And then Antoine is asking, how long does it take to distribute 10 microliters in 384 wells with the Echo? And what's the signal to count on your slide? And how do you measure that? So let's take them one at a time. How long does it take to distribute 10 microliters in 384 wells?
That's going to vary based upon what fluid type you've got. I did, as a rough estimate, my 4 microliter reactions took around 10 minutes, which is about, and that's for 4 components, it's about 1.5 seconds per reaction. So you could scale that up for 10 microliters as well. I don't know if that's what you guys are seeing as well. But.
Right, yeah, no, I think those are about the time frames. When it switches from liquid, it takes some time, but when it's shooting out, it's fast, yeah. So.
Great, and then what's the signal to count on your slide?
How do you measure that? Which slide are you referring to?
I think she's referring, or he is referring to the slide with the dots, with the photograph of the plate that you had on it. The one currently showing. Okay.
Right. Okay. This is just so these are kind of the what we're just showing here is that you can detect the products from your cytochrome P450 reaction. And show that cytochrome P450 is active and making different types of products that you'd expect. So signal counts, I think, is just arbitrary units. To help us with our process, and Ahmed Qureshi, our head of business development operations. In terms of, we have an advisory board of a lot of familiar faces kind of in the cell-free arena to really help us grow the company. And otherwise, company, we're still pretty small. I do think it's a testament that we bought an Echo as our first instrument, kind of how useful it is. But yeah, otherwise, if you'd like to learn a bit more about our technology and cell-free in general, the Medium post is out there. There's also a really nice Economist article that came out about more than a year ago now that kind of talks about technology and its potential. But with that, we're based in Berkeley, so definitely please reach out if you're ever here, and love to share around.
Great, thank you, Zach. And when did you go through Y Combinator?
We never went through Y Combinator. You're probably looking at 50 years.
Oh, I'm sorry.
Yeah, they're one of our really great early venture support.
Great, excellent. We have another question from Jason Vincent. Jason, I think you're at Syngenta in the UK, if I remember rightly. And Jason asks, what is the most complex biosynthetic gene cluster that Tierra has successfully expressed in cell-free?
It's a good question. So we are pretty focused on our kind of proof of concept right now. That's a gene cluster of 3. But you can get much more complicated than that. I think in the literature, there's increasing kind of papers that have come out expressing more and more complicated stuff. So, but again, I think an open question right in the research area.
Wonderful. Okay. So keep asking the questions if you've got them, and we'll keep answering them. Maybe you want to just unshare your screen.
Yeah.
Absolutely.
Okay.
Great. So we're going to go into the Q&A part of the webinar. I want to know in particular, talk about reagent volumes and talk about cost savings from what you would be, how much you'd be spending, and what are the most expensive reagents, and then how does the Echo change those dynamics?
Right. So, I mean, in terms of our reagents, the costs are twofold, right? One is in cell-free systems that we're able to make, and then second is in DNA that we feed into them. And both of these, by miniaturization of the assays, we can save on both ends. And so that's really big for us.
So on DNA and on reagents?
Exactly,
okay.
Yeah.
And you're making your own reagents, or you're...
Right. We make our own reagents. And so for us, right, the less reagents we have to make, obviously, the better. So that's really helpful.
And you just in E. coli, or are you doing other
different systems?
Okay.
And so that's key, right? I think the key kind of innovation that we've built out in-house is a lot of different systems that are not just E. coli based, but are active cell-free systems, right? But are not. They kind of span the gamut of diversity space.
Got it. Okay. And are you just in bacteria, or have you gone into?
Oh no, outside of bacteria.
Okay. So I remember the rabbit reticulocyte. That's a particular one that's used in mammalian.
Right. That's an older one. Right. Yeah. So rabbit reticulocyte's available. There's a bunch of different cell-free systems that are out there. Right. So rabbit reticulocyte. There's insect cell-free systems. There's HeLa cell-free systems. Streptomyces cell-free systems, and a lot of commercially available too. Yeah.
Got it. And so this TXTL is just a general.
It is, yeah.
For any cell-free system.
Right.
Translation, transcription.
Right.
Transcription, translation.
Transcription, translation. Yeah. But I mean, I think one of my TXTL from Tierra Biosciences is a great example of a really good system where it's you get these cell-free systems that they're not necessarily built for synthetic biology applications, right? So it might be just make protein and kind of use protein downstream. But MyTXTL is a great example of a system that's really been built for synthetic biology. So you can then use that and kind of build circuits, right? Start to engineering pathways, and that's a little bit of a different application.
Got it. Yeah. And I presume that came out of Ann Arbor.
That is Tierra Biosciences, right?
Yeah, Ann Arbor. Yes.
Okay.
Yes.
Great. So Jared, any additional thoughts in terms of reagent volumes and cost savings?
Yes, I mean, in the DNA assembly part, as you mentioned, we've done quite a bit of work with that as well. Sourcing your DNA can actually be quite expensive as well. That's probably, if you're making your own reagents, you're probably not making your own DNA still. So
that's right.
And we have a lot of the manufacturers of that DNA come in Echo compatible plates. So now you can actually order your linear DNA pieces that you're going to feed into your assemblies in an Echo plate as well. And I've worked with that as well on another tech note, but yeah, that makes it a lot more enabled as well. So you're able to pick what DNA you're going to put together from an Echo plate.
So did I understand correctly, some of your reagent suppliers will use Echo plates and then ship directly to you in an Echo plate?
Yes, that's becoming more common now, actually, especially with the DNA.
Very interesting. So can you rattle off some of the suppliers who do that?
IDT and Twist both do that.
Got it. On the DNA front?
Yes.
And any other suppliers in other reagents?
No, we're working on that now, actually. That's a hot issue for us, but that's a specifically good one because where the Echo is so good at hit picking and arraying, that's all that it is for DNA. when you're putting DNA together that is all hit picking and cherry picking, and that's what we really excel at. So
Excellent.
we're happy to have that. That was the first one we identified. So
great, excellent. Another question, what's the cost and has it changed with different organisms and different enzymes?
What's the cost and has it changed for different organisms with enzymes? Right, so published cost for E. coli depends on, if you're making it or you're buying it. I'm not sure what, my TXL sells it? I'm not
I can't even remember exactly. And it's
Yeah.
hard to say because everybody gets at a different price. So it's always, we always have a challenge. We were able to get a 6-fold reduction in volume. So
right.
whatever you see, divide by 6 for us. That's sort of the...
Yeah, I mean, for our systems, our other systems they're not necessarily, we're not looking to, market them. And so I think cost is less of a relevant point. But with that said, I think miniaturizing those, reactions saves on reaction volume. And so that's pretty critical for us.
And you put up what it would have cost you if you weren't using the Echo, which was what, $100?
It would cost, well, I think if you don't use the Echo, it's more, you can't really get the accuracy that you need, right? And so we did try traditional liquid handling for a while, but it's just the accuracy, you just can't really beat like 25 nanoliter, minimum volumes versus 1 microliter. It's even hard for a liquid handler to do well. So, yeah.
Yeah, and one of the best, actually, it's a little mentioned feature actually, but the Echo actually lets you know. It has an exceptions report at the end, which when you're putting together DNA, or when this is sort of a little bit of a sidetrack, but when you're putting together DNA or when you're doing these TXTL reactions, if there, as it measured that height in the liquid, if there's not enough height to actually transfer, it won't transfer and will let you know at the end. So you don't make an incorrect assumption. It removes false negatives from your system, where you're not assuming that you would transfer DNA or made a transfer without having to actually done that. So it's a very handy feature that saves a lot of time.
Right.
Great. Neha Garg has a question. What is the frequency range for the transducers and does it work at resonant frequency? Maybe you can define resonant frequency.
Actually, I don't actually know what the transducer frequency. I don't actually remember that offhand. I'd have to look it up. I'm more of the in-house synthetic biologist, but resonant frequency, I think that's referring to the fact that we typically transfer out of an Echo qualified source plate so that it does not increase. You don't have cumulative waves building up. So it's a square well, and we actually are very careful on how we actually do the transfers so that we don't have cumulative wave build up, so we don't have a propagating wave like you would in the ocean. We don't want that to happen. So we're very careful to avoid that.
Got it. And a question from Antoine for you, Zach. Does the cost of the Echo make up for the cost of reagents that you are saving?
The cost of the Echo make.
In terms of that 6-fold reduction we were just.
I think the Echo. Well, again, just to stress, I think the Echo reduces the reagent need. But really, I mean, what we love about the Echo is the fact that it's consistent, and so that's just something that no other instrument can really achieve, right? So like the cost of doing experiments over and over again because you don't get consistency way outweighs the cost of reagents for us.
Got it. Yeah. So what do you think is holding cell-free back from wider adoption?
Yeah, it's a good question. I think it's not. I think we are learning more about cell-free, and it's exciting kind of see more people understand what its use cases are. I think the story that you shared, when you learned about cell-free is a good example. The hope is that, as more people work in this field and kind of start publishing, what are the restrictions of this technology? How does it work well? Where does it not work well? That we can get more of a presence here. So I think it's probably public awareness.
So we have another event coming up here in Emeryville on April 30th, and it's all about bio-benchmarking for the synthetic biology industry. And it's with a professor at Cal called Robert Leachman, and it's being hosted by Berkeley Lights. And it's all about benchmarking for strain engineering. So if you have a design, build, test, learn cycle that you're going through, we want people to share within the industry how long it takes them to go through those cycles. So it'd be great to have Labcyte there. It'd be great to have you there because clearly this piece of it is kind of its own subloop within the design, build, test cycle, where you're doing a subroutine around cell-free and then feeding it back into the engineering of the organism. So, and also Robert Leachman has a history. He's not a biologist. He's a process engineer, and he has a history of working with the semiconductor industry, looking at chip fab and how you can speed up the design build test of fabs in the 1980s. And so we're hoping that he's going to give some input to the industry and how he engineers strains as well.
Yeah, that sounds great.
So a question from Parco. Currently, scientists are trying to wake up the synthetic genes for actinomyces or actinomycetes. I'm not sure if probably actinomyces. And there are genes from metagenomes. You mentioned AI, ML, artificial intelligence, machine learning in your technology. How are those tools used in cell-free environments? Are you doing any ML?
So I think it's still pretty early days for this. But for us, right, this idea that you can collect the actual data you need to do kind of AI deep learning techniques, it's very, very hard to do that in cells, right? To actually collect enough data points, and the fact that like a cell is just not a very controlled environment, I think would make that very challenging. And we've seen some papers come out on it, but are typically not, do design build test cycle and cell, try to figure out from there. For us, it's this potential of cell-free. We're working on getting consistent reactions and lots of reactions. Then with those data sets, then we can apply kind of learning principles, right, to them. So I think that's kind of the opportunity that we see.
And then another question from Antoine: how many and which organisms do you use to make your cell-free? And we're probably hinging on the
Yeah. So now, so we have, by my count, 9 organisms in house. They range the gamut from prokaryotes, eukaryotes. Well, I cannot share which organisms exactly we have, but suffice it to say, it's enough that we can explore some interesting space.
Excellent. What do you think is the killer app for cell-free that this could make possible?
Well, I think there's a lot of apps for cell-free. In the Medium post, I kind of highlight some of the ones. But there's cell-free for detection. You can build kind of paper-based sensors for them. There's cell-free for making things on demand. You can imagine like making vaccines kind of you can dry the stuff and it's still active. So then you don't need to ship on cold temperature, add DNA, make a vaccine, kind of distribute it. There's cell-free for learning kits, and you can build like Lego-based kits for K-12 education. That's been shown. Cell-free for pathway engineering and such. For us, at Tierra, we really think, again, cell-free is really good for natural product discovery, and so that's what we're shooting for. So I'd say that's my killer app. But with that said, I think there's a lot of stuff out there that's kind of just waiting to be explored and mature.
Zach, from a business development point of view, who is your key target? And what problems do they have, and how should they interact with you if they want to work with Tierra?
Yeah. So our current kind of partnerships that we've built out are really companies that are pan-industry, right? But interested in this idea, is there genetic space that I'm missing, to make my product of interest? And that can range from, I know kind of the genetic, we've identified a bunch of genes that might be interesting to look at. To just, can you help me understand my process better? And so I think, for companies that are kind of looking to implement kind of synthetic biology workflows, we're happy to help, with a cell-free approach.
So what's your revenue model there? It's a service-based, or it's a royalty sharing down the road?
It's a partnership-based model, right? And so I think with each company, it just depends on what the needs are and what we can provide.
Okay, great. Jared, what do you think is the killer app for the Echo? Are there other things that you think are like cell-free, maybe, that are just on the cusp of becoming big, that you're seeing your early adopters start to do?
I think I think it's not just an app. I think it's entire workflows. That's sort of, as an application scientist, that's what we're trying to do. Not just dispense single reagents or single compounds, but we have an automation system that after you buy the Echo, you can buy that as well and put it on there. And you can have an entire automated workflow where you hit play, you come in the next day, and you've ran 20 plates worth of assays work. So that's sort of, I see that happening next is it's even more automated. It's even less human interaction, and that will improve consistency and also drive down cost of the whole system.
And if somebody wants to get going on the Echo, what should they do?
There's actually a website. We have a very nice website that's been put together.
It's a new website. I like it.
Yeah, it's very friendly and very nice. We have a good marketing team. They can contact us through that. They can. We have a lot of publications out there as well that we put out on that website. So if you're interested in the DNA assembly or another one of the processes, we have that and up on there. And we have plenty of salespeople that are, depending on where you actually are, they will get in touch with you. So if you get in touch with someone off the website, they can then direct you to the appropriate salesperson.
Great. And you have a system where you'll send an echo out to a lab?
Yes. If there is a need, if it's particularly a novel aspect that we haven't done before, or what we're interested in, we can do a demo on site. Actually, we have a lot of really good field application scientists that will go out and do in-house, and that's one of the things we're known for. Is actually we really are concerned with our customer base, and we are we have a lot of really smart people that work there. So they're able to help out a lot with different workflows and different tasks that may be new to the Instrumento itself, and we might have not even thought of. Our customers actually tend to bring in a lot of the interesting ideas and applications to us. So that's a good way of going forward.
And remind people, what have you done at SynBioBeta in the past in terms of the workshops that you've done there?
So we did NGS. We also did DNA assembly, and then last year I actually did this TXTL one. I did the basics for that. So I did the TXTL workflow. But NGS is a hot button issue as well. So my other coworker, my other application scientist, has quite a few papers on that, just tagmentation and actually how to miniaturize like the next Tierra XT kit and on a lot of those things. So it goes hand in hand with this whole. We're doing the read, write, and edit of DNA assembly as well. So
excellent. And if you want to find out about those applications, then you can refer to our Amaris webinar that we did a couple of months ago. So we're going to wrap up in a couple of minutes. So we'll try to take as many questions as we can before that. Sure. Dongwon Park says, are there any papers out there that use Echo to make a natural product?
That use the Echo to make a natural product? I don't know. Are there?
I've read quite a bit.
Yeah.
I'm sure there are.
I mean, maybe we talked about
Amaris is doing that. Amaris is doing that. So I mean, Amaris has done that, and they've published. Actually, they have published that. It's mainly piecemeal. Like they'll talk about actually making the DNA, the plasmid for it, or they'll talk about that kind of a thing. But it's sort of they talk about the individual components needed to make that natural product.
I mean, there have been papers on using Echo and cell-free. I think Paul Fremont's group has a paper out in 2017 or 2018. That's worth looking at. I'm not sure if they're making natural products with it. They might have, but that's a potential site. Yeah.
Great. Yeah, that's Paul Fremont at Imperial College.
That's right.
David Carrick says, I'm sold. Do you know of any good equipment grant opportunities for fellow academics? So grants so that academics could write to get equipment grants to get the Echo.
They would be able to talk to the salesperson actually, and they could point them in more in the right direction with that. I'm not much of the grant writer type, but yeah, they have our salespeople are very friendly, and they enable people to buy one. Very, very helpful.
So how should David get in contact with somebody?
The website itself will actually point them in the right direction. Our website will actually give them the contact information, and they should be able to be pointed to the correct salesperson for their location.
Right. And I'd say that I think having like access to a core too is because your typical run just doesn't take very long. So you might not even need it in house if your university has one. The core. I think that could work too, pretty well.
Great. And then a question from Wen. What is the success rate when you transfer what you learn from cell-free back to the cell?
That's a big question, I think.
Great question.
A lot of people ask that question, right? So you collect a lot of data in cell-free, but if it doesn't translate at all, then why do I care? I think there are two answers to that. One, many applications don't rely on moving it back to the cell. You can have purely cell-free applications, and so in that one, that's a little bit irrelevant. But for the second one, I think again, this is somewhere where we love to see more literature come out on the topic. In some work that I did in my PhD, we saw that you could kind of engineer oscillators starting in cell-free systems and then have them work in cell, and then different parameters like protein degradation, the kind of effects that you'd see in cell-free systems match those in cells. We've also seen that kind of promoters and UTRs, when you compare them cell-free and cells, they're not one-to-one correlations, but they're broadly applicable. And so I think transfer functions people have done too and shown they're pretty similar. And so I think with more work in this area, we'll understand more kind of the relationship between the two. For me, I don't see it as a one-to-one correlation, but I see it rather as cell-free as a way you learn about your process. You take that learning and you transfer it into a cell, and that's what's powerful about it.
Excellent. I think we have the last question from Antoine. Does the freeze-dry protocol work well with the lysate-based cell-free? Can you conserve it at room temperature?
That's a good question. There are freeze-dry protocols that have, I think in the literature, has been shown with the lysate-based stuff. And I believe it's been conserved at room temperature, but I'm not certain. I know it definitely has been done with kind of purified component type stuff, and I know that can conserve pretty well. So.
Great. So just before we wrap up, I have a final question for you.
Tell
What is the utilization rate of the Echo in your lab?
Of the Echo.
Is it running all night? Is it running every day?
So far, it's not quite running all night, but it would be great if it could run all night. So when you have those technologies, definitely let us know because we'd love those.
Awesome.
Yeah.
Great.
Good.
Any final thoughts?
I think that's good. Thank you for having us.
Absolutely.
It was great working with you all.
Yeah, this has been fun. Final thoughts, Jared?
No, it's thank you for working with us. It's great to see it being used. It's actually great to see a customer using the instrument that we're working on. So it's very nice to see a real use case.
Yeah, that's great.
Awesome. Well, thank you all for joining us. Tomorrow morning at 8 a.m., we are doing another live webinar, Introduction to Synthetic Biology. That's a 3-hour overview of the technologies, the industry, and the investment opportunities. So I hope you'll join us for that. That's 8 to 11 Pacific. And we have, as we said, a whole session on cell-free synthetic biology taking place at the conference in October, and a workshop from Labcyte that will be taking place there as well. So if you want to roll up your sleeves and get some hands-on experience, then come to the conference in October, and you can find details of that on the website too. So thank you all for joining us, and we will see you next time on the next SynBioBeta Deep Dive. Great. Thank you, guys. That was fun.
Thank you.
Yes.
Yeah.